Hello everyone, welcome to Chandu Biology classes. My name is Deepesh Joshi and today is the second lecture for our CUET PG as well as CAT-B series in which we will cover the most important topics which are important with respect to your upcoming CAT-B and CUET PG 2026 examination. And we have started with the first lecture of our series in which I have taught you about the different kind of plant pigments, their role, what is reaction center, what are antenna molecules, as well as what is the basis by which the transfer of energy happens from one pigment molecule to another pigment molecule, and the different kind of pigments which are present and all these kinds of things. And we have also looked over into few PYQs. Okay, so this is the continuation of that lecture in the second part, in which I will be dealing in detail about the photosystem 1 components. So we will discuss that what is the photosystem 1 made up of. Okay, so in the second lecture, and also we will be moving towards the inhibitors of photosystem 1 from which the questions usually come in the exam, and we will solve a good number of questions today as well. As in this lecture I will be only dealing with photosystem 1, so we will solve some questions on it, and then in the another lecture we will be focusing on photosystem 2, and then we will finally go with cyclic and non-cyclic photophosphorylation. So my name is Deepesh Joshi. As I have told you, I am an integrated PhD scholar at ISC Bangalore, and I have qualified all these examinations. So basically, I will be focusing more today on the CUET PG as well as your GAT-B exam. Okay, so on basis of that only, I will be covering all the topics. So let's begin with our lecture. So yeah, so I told you about the light harvesting complexes. Okay, these light harvesting complexes are the crucial part of a particular photosystem. So basically I told you that you have something you called as a reaction center. Okay, that is at its lowest energy and highest wavelength. Okay, its wavelength is highest, that means it can absorb the maximum wavelength as compared to another molecules which are present in the entire photosystem, which are nothing but these are your light harvesting complex or antenna molecules. Okay, so if you compare with respect to them, they absorb at comparatively lower wavelength. Okay, so basically your reaction center, these are of two types. One is your PS1, another is PS2. PS1 is also called as P700 because the maximum absorption range for the specific wavelength at which the reaction center of PS1 absorbs the light, that is 700 nanometers, and for PS2 it is 680 nanometers, so it is also called as P680. So this is the reaction center, and these are the light harvesting complex or antenna molecules. So basically this reaction center is composed of your chlorophyll A in case of your higher plants. So basically if you see that the light harvesting complex and chlorophyll AB, they form the antenna protein complex. Okay, and you have different photosynthetic pigments like chlorophyll A, chlorophyll B, carotene, xanthophyll. So all these chlorophyll B, carotene, xanthophyll, they are part of the light harvesting complex, and chlorophyll A is something that is part of the reaction center. Okay, so this is something that you should know. And the protein complexes, now we will be dealing with the protein complexes. So basically if you talk about the protein complexes, it is composed of two kinds. One is a LHC1, that is a light harvesting complex 1, and the second one is LHC2, that is light harvesting complex 2. So LHC1, it is a dimer. Okay, it is dimeric in nature. It is composed of two subunits, and the molecular weight of this is 50 kilodaltons. Okay, that is the molecular weight of the whole dimer. So on the basis of simple unitary method, what you can tell me that each subunit is having a molecular weight of 25 kilodaltons, and the LHC2 is a trimeric molecule. It has an overall weight of 75 kilodaltons, so each subunit will again have a molecular weight of 25 kilodaltons. And the LHC1 is encoded by the gene that is called as LHCA, and this LHC2 is encoded by the gene that is called as LHCB. And you always write the genes in small letters. Make sure of this. You write proteins with capital letters. Okay. So if you have to write the protein that is responsible, then you will write in caps. Okay. So that is how you are having these genes which code for this LHC1 as well as this LHC2. Okay. And LHC1 is dimer, LHC2 is trimer. Okay. So just remember with me. So how you can remember which is dimer, which is trimer? One simple trick can be that it is LHC1. After 1 in numbers line, you get 2. That means LHC1 is a dimer, and LHC2 after 2 in the number line, you get the number 3. So that means LHC2 will be trimeric. So that is a short trick to remember these things. And guys, if we move further, so we can see that the photosystem 1 of which we were talking about, that is nothing but your P700. It is composed of a variety of protein complexes. So basically you are having in this red color, you are having this PSA-A, then you have this PSA-B, and surrounded by the PSA-A and PSA-B, you have something you called as a PSA-C protein that is again associated with the A2 subunit of this photosystem 1. So basically in this photosystem 1 you have a variety of carrier molecules. What are carrier molecules? Carrier molecules are those molecules which can accept electrons and they can transmit the electrons also. So that means they can act as a channel for the transport of electrons. Okay, and as these electrons flow from one channel to another channel or one protein complex to another protein complex, there is always a downhill movement that is happening. Downhill movement means that it is moving from the tendency of higher reduction potential. That is the what is reduction potential, guys? I hope you know this much chemistry. Reduction potential is basically the tendency to accept an electron pair or a given electrons you can accept. So that tendency is measured in terms of reduction potential. So basically the movement in the entire photosystem, in the entire cyclic or non-cyclic photophosphorylation, it happens from the higher reduction potential to lower reduction potential. This is something that you should know. So like that the movement happens. So basically then this photosystem 1 is also surrounded by this LHC1 proteins. Okay, this LHC1. So I told you that LHC1 is nothing, it is dimeric. Okay, it is dimeric. So you can see there are two purple LHC1 which surrounds this photosystem 1, and the photosystem 1 is also surrounded by these proteins which are PSA A, B and C. Okay, and within this photosystem 1 you are having some different components, like you have this A0, A1, A2. What is A0, what is A1, what is A2? Very important question, guys, for the examination point of view. So basically this A0 is referred to as chlorophyll A. A1 is nothing but phylloquinone, and A2 is iron sulfur protein cluster. So that iron sulfur protein cluster, it consists of 3 proteins. Okay, it consists of 3 proteins which leads to the transfer of electrons, and this A2 is associated with PSA-C protein. That is something you should remember, that the iron sulfur apoprotein that is associated with PSA-C protein, this is being coded by the genes. So here I have written the genes. So basically PSA-C gene, it codes for PSA-C protein, and that is associated with iron sulfur apoprotein, and PSA-A is gene is encoding the PSA-A protein, and PSA-B gene is encoding the PSA-B protein, which is a part of reaction center. Very important, that for reaction center to form the core of the photosystem, you are having this PSA-A as well as PSA-B. Okay, so this is the thylakoid membrane, and in the membrane only, like this part is your stroma, this part is your lumen. So you can see that the PSA-C that is present towards the stromal side. So on the location of the proteins also you can get some questions. And as soon as some light of specific wavelength. Most probably it would be 700 nanometers. As it falls, this electron flow happens from A naught to A1, then to A2, and then there is a lot of other things which are happening, which we will deal with in cyclic and non-cyclic photophosphorylation. Till then, just hold your excitement and just understand this part, that is the structure of the photosystem 1. So I hope this structure is clear to you. What is A naught, what is A1, what is A2. Okay, if all these things are clear, so basically we can move further. So if we talk about the photosynthesis, so the photosynthesis mainly it can be oxygenic photosynthesis, okay, and it can also be non-oxygenic photosynthesis. In case of oxygenic photosynthesis, there is a evolution, or there is a formation of O2 gas, okay. But in case of non-oxygenic photosynthesis, your oxygen is not produced but other compounds are produced, okay. So here in case of oxygen, there is a photolysis of PS2 as well as PS1, and mainly oxygenic photosynthesis. Basically, it happens in green. It is found in green plants, green algae, and cyanobacteria. But the non-oxygenic photosynthesis, something that is you called as a photolysis of H2S is happening over here. Okay, and it is found in the purple bacteria, and the reaction center in purple bacteria, basically it's also called as P865 because it absorbs the light of 865 nanometers, which is above the red region, like the far red region, okay. And the green sulfur bacteria, it is the reaction center is P840 as it absorbs 840 nanometers of light. So these are the examples of purple sulfur and green sulfur bacteria. So in the CUET exam you can get a very simple question out of this slide, that purple and green sulfur bacteria, they show oxygenic photosynthesis, non-oxygenic photosynthesis, no photosynthesis, or none of the above. Your answer should be non-oxygenic photosynthesis, okay. So I hope this part is clear. Then here you can see the different structures. Basically, this is the diagrammatic representation for the reaction center, okay, and the bacteriochlorophyll dimers. And there is a homology between the reaction center, that you can see that here you have in bacteria you are having this P865, okay. In the purple sulfur bacteria, then you have these two units M and L. Then in photosystem 2 you have these units which is D2 and D1, that I will deal with in detail in the lecture 3, okay. So this we will cover in lecture 3, the detailed structure of photosystem 2. Then you can have this photosystem 1, that is P700, okay, in which you are having this A or B subunits, okay. So basically this is the overall structure similarity. And then if you see it further, there is a phenomena that is called as photooxidation, okay. This photooxidation is very important phenomena. So what happens in photooxidation, let me tell you. Let us zoom this flow chart and understand everything in detail, okay. So basically what happens is in tropical conditions, okay, in tropical conditions, when you are having a very high light intensity, so the intensity of light is very high. So when a high light intensity falls on the plant, then the fate of the absorbed light energy, it basically depends on two line of defenses. So basically in our body, like if you encounter an infection, okay, so then you are having different immune soldiers, and these soldiers, basically you are having these different layers of protection, okay. So you can have the first line of defense, that is the primary protection, and you can have secondary protection as well, okay. So if the primary protection is not sufficient, then the secondary protection or the second line of defense plays a crucial role. So basically, primarily if a plant is being irradiated with a high light intensity, then that plant can have some sort of mechanisms by which it can protect itself. So that mechanism can be thermal dissipation. The plant can absorb the light energy that is falling on it, and it can dissipate the heat that is produced, and by dissipating the heat it is protecting itself from high light intensity. So that is one mechanism. Another is something we call as a xanthophil cycle. Let's not go into detail of xanthophil cycle because that is not important from your exam point of view, but it is another first line of defense. That is something you have to remember. And the next, it can be non-photochemical quenching. Okay, so something you call as a non-photochemical quenching. So in non-photochemical quenching, you can have, like, you can do photosynthesis. Okay, by photosynthesis you can utilize the light energy and you can protect yourself. Then in case if the excess light is not fully dissipated by the three mechanisms, then you can have formation of some different states of chlorophyll. Okay, that chlorophyll, basically you have this triplet state of chlorophyll that can be formed, and there can be generation of reactive oxygen species. Okay, so if the ROS is being generated, okay, and your chlorophyll is attaining a triplet state by absorbing the light energy, then that chlorophyll molecule, basically you need some kind of mechanism of protection that is called as photoinhibition, in which there is a D1 protein that is present in the PS2. It is degraded, and with the degradation of the D1 protein you can encounter such things. So when I will be dealing with PS2, then I will be dealing in detail with the D1 protein degradation and the pathways, and we will come back to this point. Okay, and in the second line of defense you can also have a antioxidant system. So basically in case of antioxidant system you can have different mechanisms. The first mechanism, it can be, you can have a presence of scavenging molecules. Scavenging molecules are those molecules which can reduce your burden by taking it. So what are these molecules? These are nothing, we have studied it, in plant pigments. So basically beta carotene. Okay, and you can also have ascorbic acid, vitamin C, tocopherol, superoxide dismutase, reduced glutathione, and anthocyanins. So these pigments, beta carotene, anthocyanins, these we have looked in the plant the first lecture, in which we have seen that the antenna molecules which are there, which makes up the light harvesting complex, it is composed of this chlorophyll B, carotenes, xanthophylls, and all these kind of molecules. Okay, so these are the scavenging molecules which can lead to protection of plant from photooxidation. And another thing is you can do ROS neutralization by some of the proteins, and you can prevent the oxidative stress. So this is the overall thing about photooxidation. Main thing which you have to learn is that your light harvesting complex, basically it has two functions. Okay, the light harvesting complex, it protects from photo oxidation, and it transfers light to reaction center. So these are the two important functions of the light harvesting complex. Okay, so if the photo oxidation part is clear, the D1 degradation I will tell you. Now we can look over to some of the inhibitors of this PS1. So this PS1, it can have some sort of inhibitors. Some chemical species can inhibit the PS1. So what are those chemical species and how can they do it? So basically you have something that is called as a paraquat. Paraquat is a methyl villogen. That is a compound you can see over here. It has in this linkage N double bond N with followed by the ring, and there are two methyl groups which are present. So basically this compound, it accepts electron from early acceptors of PS1, okay, and finally reacts with oxygen to form superoxide that damage specifically the lipid component of chloroplast. So what does this paraquat does? So basically if you look over here, this is your entire PS2 PS1 complexes. So PS2, it is associated with oxygen evolving complex, okay, or you can say manganese associated complex. This oxygen evolving complex, it can lead to photolysis of water, and then if 680 nanometer wavelength less than 680 nanometer is being irradiated, then the antenna molecules absorb it, transfer it to the reaction center, flow of electron happens, and as soon as the electron flow reaches PS1, from PS1 basically the electrons, when they are passed on to this ferrodoxin, so in between this paraquat comes into the picture. It captures the electron, and by capturing the electron basically it leads to the formation of reactive oxygen species. This reactive oxygen species, basically the superoxides which are there, the superoxides, they can lead to damage in the lipid component of the chloroplast, and the, what do you say, the FNR, that is the ferredoxin reductase, it does not get any electron to produce NADPH from NAD plus. Okay, so NAD plus to NADPH, this process is being now halted or inhibited, and this paraquat captures the electron, produces ROS, and damages the chlorophyll chloroplast. Okay, so basically this is an important inhibitor of PS1, that is something you have to remember. And similarly, like you can have other inhibitors also, like for plastoquinone. So we will deal with plastoquinone in the subsequent lecture. So basically it has some inhibitor called as DBMIB, so I will deal with it, don't worry. And if this much part is clear for today about the photosystem 1, the structure, the components, then we can solve some questions to better understand our topic. So the question, if we start with, is the first question, the one of the most widely used herbicide, methyl viologen, that is paraquat, interfere with what? It interfere with the photosynthesis of higher plants, and they are responsible for evolution of more oxygen. So guys, tell me fast, okay? Whenever there is a question, we have to solve it within a minute, okay? Because CUET and GATB questions, they are like really easy to solve. You have to read the question, you have to understand the fact, you have to process it very fastly. For that you need effective revision. Okay, so basically if you have this, so here in case of herbicide, so basically it is asking that when these herbicides like the paraquat, it is inhibiting your photosystem 1, then what it is happening, what it is causing, what are the causation. Okay, what is the causation? So basically evolution of more oxygen, does it happen? Does I told you anything to do with the oxygen? Because oxygen evolution is associated with PS2 and that is functioning properly, so this might be the wrong answer. Dissipation of proton gradient across thylakoid membrane, there is no change in the proton gradient, that is also false. Inhibition of full flow of electron from PS2 to PS1, as I told you in this diagram, when I taught you, I told you that the electron flow happens, and as soon as it reaches PS1, there is no, you can say, problem, or there is no like there is no problem in reaching to PS1. After PS1 there is a problem. Okay, so basically this is wrong. Transfer of electrons from PS1 ferredoxin to molecular oxygen to form ROS. Okay, so basically this is the thing that is happening when you are using paraquat. That is, this is forming reactive oxygen species. Okay, so basically this is something that you need to remember. And coming over to the next question, based on the structure that we have studied, which of the following electron carriers of photosystem 1 is mobile? So as I told you that mobile carriers, okay, basically here you are having different kind of iron sulfur proteins, A0, A1, A2, okay, all these kinds of things. Out of them, this ferrodoxin, okay, this ferrodoxin, it is the mobile carrier. Okay, so this is something that you should remember as a fact, that the ferrodoxin, that is the mobile electron carrier. Okay, then moving over to the next question, basically the reaction center P700 of photosystem 1 is composed of. So what does the reaction center of photosystem 1 is composed of? two chlorophyll A molecules, one chlorophyll A and one chlorophyll B. So there is no chlorophyll B, it is present in the antenna molecules. One chlorophyll A and one pheophytin. No, I told you what is pheophytin in the previous lecture. No, that is wrong. Two chlorophyll B molecules, wrong. It consists of two chlorophyll A molecules. Chlorophyll A, it forms the reaction center, and there are two chlorophyll A molecules which are present in the reaction center of PS1, that is P700. Okay, so this is something that is important. So the correct answer is A. Next question, guys. The photosystem 1 is primarily located in which region of the thylakoid membrane? So I told you this in the previous lecture also. Let me told you again. So basically these are your thylakoids which are present. Okay. So in the thylakoids there is some part that is exposed to the stroma. Okay. The part that is exposed to the stroma, the terminal thylakoids mainly. Okay. Or these thylakoids which are there, which are present like this. So basically the ones which are exposed to the stroma, they are being called as non-oppressed or stromal thylakoids. Okay, and the thylakoids which are sandwiched by the neighboring thylakoids, they are being referred to as oppressed, or you can say them non-stromal thylakoids. So basically photosystem 1, that is primarily present, okay, in your which kind of thylakoids? Okay, so are they present in oppressed granar region, non-oppressed stromal lamellae? This is the correct answer, because here in the stromal thylakoids you have primarily the PS1. PS2 is absent in stromal lamellae or the stromal thylakoids. But in case of your non-stromal thylakoids, you are having both. You have PS1, you have PS2. So there is no primarily something that is located. Primarily PS1 is located only in the stromal thylakoids. Okay. So that's why the correct answer is B. Now coming over to the next question. Which of the following is the first electron acceptor structurally associated with a reaction center of photosystem 1? So if you remember the diagram, I told you it is A0 to A1 to A2. That means what is this A0? A0 is nothing but chlorophyll A. So the answer is C. Okay, answer is C. Chlorophyll A, which is also written as A0. Okay, very good everyone. Let's move to the next question. The iron sulfur centers FX, FA, FP. So I told you that the iron sulfur basically of photosystem 1, it is made up of three proteins, the iron sulfur centers, that is FX, FA and FB. So these are bound to which protein subunit? Okay, you have to tell the name of the subunit. So I told you about the different subunits, correct? So out of these you can see over here in the initial slide, I told you that the iron sulfur apoprotein is associated with PSA-C, and PSA-A and B, they form the reaction center proteins. So basically if this is the case, yeah, in this question, if this is the case, then our answer should be PSA-C, that is B. Okay, B option. So coming over to the next question, guys, if we can see that which pair of subunits forms the core of the photosystem 1 and binds most of the chlorophyll molecules. So I told you that the PSA-A and PSA-B, they form the core. Okay, so basically answer is B. Okay, so you are also having different PSA, but for your examination point of view, PSA-A, B, C and D also, like D is associated with the ferrodoxin. Okay, so that is something that you can remember, but PSA-A, B and C, these are the most important ones. Okay, now next is how many iron sulfur centers are present in photosystem 1? How many iron sulfur centers? So I told you that there are 3 iron sulfur centers which are present. So answer is 3. Okay. And then you have this question that phyloquinone, that is A1 in photosystem 1, is structurally equivalent to which type of molecule? Okay, so phyloquinone, with the name only you can tell guys that phyloquinone will be equivalent to quinone. Okay, so this is a modified form of the quinone, so that is why your answer will be here B. Okay, answer is B. So then the next question, which photosystem 1 subunit provides the docking site of ferredoxin? So basically ferredoxin, it is present more towards the stromal side. Okay, and here you were having this PSA-C, and then here you were having the PSA-A and B. Okay, so this is obvious that this ferredoxin, since it is present on the stromal side, then it will be associated with PSA-D. Okay, so that is why answer is D option. Wait, the answer is D. Okay, so yeah, and then the next question, the photosystem 1 core complex typically contains approximately how many chlorophyll molecules? Okay, so basically the number of chlorophyll molecules are 100 to 120. This varies from plant to plant, but technically they vary from 100 to 120. Okay, so please note the fact. And yeah, so we are done with this lecture about photosystem 1. We have looked over to the structure, to the inhibitor of PS1, as well as photooxidation, some basics. This D1 protein degradation I will tell in detail in the next lecture, and all these things we have covered. So yeah, so thanks for joining the lecture, and let's meet in the next lecture.